So now we're going to be looking at, in this tutorial, at dihybrid crosses, which for a dihybrid cross is a specific kind of a two-trait genetic cross. And over here we see how we might go about getting a dihybrid. If we start with a true breeding yellow and round peas, which are the two traits, the color and the texture, and true breeding green wrinkled, and we cross those with genetically cross them, then we're going to get our hybrid here. So our hybrid is shown to be both yellow and smooth. So this indicates that the smooth texture and the yellow color are the dominant forms. So overall, then that would make our genotype for this for round, R for round, and the little r would be for wrinkled, and then big Y for yellow, and the little y for green. And so this would be the genotypes for all the hybrids. So if we were to take this hybrid and breed it with another hybrid, that would be a dihybrid cross. And so how do we predict what the ratios of the offspring would be the question. But the first thing we need to do is determine what kind of gametes can be produced when we consider two different traits now. So that's what this one is about. We're going to learn to determine the number of possible allele combinations during gamete formation, gametogenesis, which involves meiosis. So one of the big ideas here is indicated by the first bullet. It says that if alleles for the two different genes do not influence one another, and that is when they're being separated during meiosis, then the alleles for the different genes, in this case we're doing two, are going to be separated or segregated into gametes independent of one another. So that means as we separate the alleles for the first gene, whichever gametes they end up in does not influence the second gene and the two alleles from there. So that means we need a strategy for determining what are the possible gametes that we can get. So here's a little practice here. And the first thing we want to do is just see how many possible gametes can an individual or can individuals with the following genotypes produce. That should be just can individuals. And so let's say we have the following genotype. This is just generic, gene A and gene B, and you see it's double heterozygote, so heterozygote for first and the other. And so if we look at the first gene, AB, however they segregate into the gametes during gamete formation is not going to influence the second one. So that means that if one of the possibilities is to separate and get a gamete that has big A, big B in it. So remember, we have to separate both. We have to separate the A's into gametes. We have to separate the B into those same gametes. So one possibility would be to get big A, big B. The next possibility is to get big A and little b. So big A can go into one gamete and the little b can go into that gamete. So that would be another possibility, big A, little b. And then another possibility would be little a, big B. And because remember, gametes are only supposed to get one copy of each gene because you only get one complete set of chromosomes in your gametes. And then finally, the last possibility is little a, little b. So we were kind of following along, kind of followed a little strategy here. We did the first one for each gene, for each trait. That would be big A, big B. So that was the first one. Then I did the outer ones. Okay, so that would be big A, little b. So that's the outer ones. And then I did the inner ones, little a, big B, and then I did the last. Kind of like in algebra when we were doing the FOIL method, and that's a good way to come up with them. So how many possible gametes can we get? Well, it depends on the types of combinations with those alleles, and that's a total of 4. So there's a quick way to do it here. Because this one does not separate, does not influence, the A gene does not influence how the B gene alleles segregate, then as far as that goes, that's a multiplication rule. So You can either get a gamete with a big A or a little a, so that's 2 possibilities. And then you can get a gamete with either a big B or a little b, and that's 2 possibilities, so that's 2 times 2, and that gives us 4. So let's try that here. On the second example, how many possible gametes can we get, which is actually the first question here. Well, you can either get a big A or a little a, so that's 2 possibilities there. But for the second gene, there's only big B, so you can only get 1 possibility there. So the total possible gametes that you can get is 2 times 1, which is 2. So, and if we tried the FOIL on this one here, we would see that would be the case. If we do the first 2, we're going to get big A, big B. If we do the outer, we get big A, big B. If we do the inner, we're going to get little a, big B. And if we do the last, it's going to be big A, little a, big B. And so if we look and think about it, these 2 are the same, so that's only 1 type. And these 2 are the same, so that's only 2 types, so we're going to get 2 types there. And now we come to a little more complex situation where we now have 3 alleles. And trying to figure out the actual combinations would be a challenge, so we're not going to do that. We just want to know how many possible gametes. And this is misspelled here, so how many possible gametes can individuals with these genotypes produce? So here we can get either a big A or a little a, so that's 2. A big B or a little b, so that's times 2 because they don't influence each other, so it's a multiplication rule. And then a big C, a little c, that's times 2. So the total number of possible gametes is 2 times 2 is 4, times 2 would be 8 possible gametes. And so how many do you think you can get on this last one? And now that's 4 traits. Gene A, Gene B, Gene C, and Gene D. And so let's think about that for a little bit. And if you came up with 8 on this one, you would be correct. The reason for that is it's 2 for the first one for A because it's either big A or little b, but you have big B and big B for the second one. That's only 1 possibility, so that's 2 times 1. Here you have 2 possibilities for C and 2 possibilities for D, so that's 2 times 1 is 2, times 2 is 4, times 2 is 8. So here, in this case, even though we have 4 different traits that we have alleles for, there's only 8 possibilities there because one of the genes or one of the traits is homozygous, so there's only 1 possibility for the B gene. So now let's look at an actual genetics problem, and we ask the question, what is the expected phenotypic ratio for a dihybrid cross for P texture and P color? And the alleles are defined for you here, so it looks like capital R means the round is dominant, little r wrinkled is recessive, color yellow is dominant, and green is recessive. And so if we're doing a dihybrid, that means you've got to be heterozygote for both. Anything other than that, and you're not going to be a dihybrid cross. So what would the cross look like? It's going to be basically big R, big Y, little y. So there's your first hybrid, and we're crossing with another hybrid. Okay. So if you don't have 2 individuals that are hybrid, they're heterozygote for both, and it's not a dihybrid cross, it would just be a 2-trait cross. So I'll give you an example here. If we have one individual that has this genotype here, and this could be a whole other genetics problem to work using a Punnett Square. This one is technically not a dihybrid because the first individual is not dihybrid. So this is just a 2-trait cross. So if a problem ever tells you dihybrid is very specific and both genotypes, you're heterozygote for both traits. Okay, so we're not going to do this 2-trait. We're going to do the dihybrid cross, and we're asking what is the expected phenotypic ratio. So we're going to need, for the first individual, we're going to need a Punnett Square that is built to reflect the number of possible gametes. So remember, there's 2 possibilities for R and 2 possibilities, so that's 4. So we're going to need a Punnett Square for the first individual that's going to have 4 columns, so that we can put the alleles for each gamete there, possible gamete. And the second one the same, so you're going to need 4 rows there. Okay. And if we use that strategy, the first, outer, inner, last strategy to figure out the gametes, the first one is big R, big Y. The second one is the outer ones, big R, little y, and then the inner, little r, big Y, and then the last is little r, little y. And then you're going to have the same for the second one because it's the same kind of genotype. So big R, big Y, big R, little y, little r, big Y, and little r, little y. And so now what the Punnett Square does is allow you to figure out what the possible genotypes are, and from there we can figure out what the possible phenotypes are because we're being asked for the phenotypic ratio. So all we do is fill in all the possible fertilizations that can occur from the types of gametes produced. And I'm going to go ahead and do that right now. If you're following along and doing this yourself, you can work on filling them in too. So now that we filled in all the boxes, we're going to want to figure out the ratio for the possible genotypes for this dihybrid cross. And so what we're going to do is we're going to start with the first box here and figure out what that genotype is. And that genotype looks like it's going to be round and yellow. And so now what we want to do is go and figure out how many other round and yellows there are. So we know we already have one. The second box is going to be round and yellow too, because we have at least one big R and big Y for the genotype. This one is also round and yellow, round and yellow, round and yellow. This one is not. The second one on the second row. This one is round and yellow. This one is not. This one is round and yellow, round and yellow, no, no. And this last one down here on the bottom left is round and yellow. And if we scan, the rest of them are not round and yellow. So how many is that altogether? 1, 2, 3, 4, 5, 6, 7, 8, 9. So a total of 9 round and yellow. So now we're going to look for the very next one and figure out what this possible genotype is. And that's the second box on the second row, and that looks like it's going to be round and green. And so that's one, round and green. That's another one that's going to be round and green. That's not round and green, no. This one down here, round and green, not round and green, not round and green. So that's a total of 3 round and greens. So now we look at the third row on the third box, and there we see a wrinkled yellow. So that's something different that we don't have yet. So we've got wrinkled and yellow, and let's see how many we've got there. So here's one wrinkled yellow, 2, here's another one, 3, and the last one is not. So that's 3 wrinkled and yellow, and that leaves the last one, which looks like it's wrinkled and green. And so that would be 1. And if we add all of these up, that's going to add up to a total of 16. So we expect then that 9 out of every 16, the ratio of 9/16 in a dihybrid cross would show the full dominant, and the last one would show recessive for both, would be only 1/16. And the middle two are going to be 3/16 each, and we're going to refer to these as recombinants, because if we went back to how these hybrids were produced, the parental generation that gave rise to these F1 hybrids would have been pure breeding, round and yellow, crossed with, so this would have been the parental generation, okay? And they would have crossed with pure or true breeding recessives, which would be homozygous recessive for wrinkled and homozygous recessive for green. And so these parental types, that would be both round and yellow, which is exactly like the first group here, round and yellow. And so that the phenotypically, that's like the parental type. And the second parent matches with the last group or category of phenotype, possible phenotypes, wrinkled and green, where both are recessive, and that matches the other parent from the parent generation. So what do we then have now is what we call recombinants, because they are different combinations of the original parent generation. In the original parent generation, we did not have a round and green, we had round and yellow. And in the original, we didn't have a wrinkled yellow, but we had wrinkled and green. So these represent a reshuffling of those two traits to get two recombinants. So the middle group's always going to be recombinant. This is something to remember, anytime that we have a dihybrid cross and the alleles separate independent of one another for each of the traits, then you can always expect a 9:3 to 3:1 phenotypic ratio, where the first one is dominant for both, second or the last one is recessive for both, and the middle two groups are the recombinants. And that's something to remember because it'll help you answer a lot of genetics questions. So now let's take a look at another two-trait cross, and this one does not appear to be a dihybrid, but it's still a two-trait cross, and we can still use Punnett Square to solve this one, and we can probably make things a little bit easier on ourselves in cases where we're homozygote for a particular trait. So here we're looking at a purple flower tall pea plant, and this is its genotype. It's homozygous for purple and heterozygote for tall, and that tells you that purple, because they're capital letters, is the dominant and tall is the dominant, but heterozygote in this case. And it's going to be crossed with a purple flower, and in this case this individual's genotype shows heterozygote for purple, and the plant is short, so short is recessive, and we're homozygous recessive there. So it's always helpful to write out, I find it helpful to write out what the cross looks like so I can see it, and then I can build my Punnett square from there. So that's the first parent, who's purple and tall, and the second parent is also purple but short. Okay? And so we can see here that for the Punnett square, there's really only one kind of P you can get, so it's 1, and then there's 2 kinds of T's you can get, so that's 2. So really we're going to need a Punnett square that has only 2 rows or columns for this parent, and the same goes for the other one because it's 2 and then 1. But let me show you. We'll go ahead and build the entire Punnett square using the first, outer, inner, last, and then see why we really don't need a 4x4 Punnett square. Okay, so I'm going to draw my 4 columns and then my 4 rows, and we'll go with the first parent on top. If I do the first, that's big P, big T. If I do the outer, that's big T, little t. If I do the inner, that's big P, big T. And if I do the last, that's big P, little t. And if we look right here, we see that this one and this one are the same. The first one and the second one, and then the second one and the last one are the same. And it turns out that when we run the entire Punnett square and we get all of our results out of 16 boxes, the ratios actually reduce overall to where we didn't even have to put the column that had that same set of alleles in there. But I'll show you here in a bit. So here's the second parent. So if we do the first, that would be big P, little t. If we do the outers, that would be again big P, little t. And if we do the inners, that would be little p, little t, and the last big P, little t. And again, we only really have 2 types of gametes. So if we went in and we filled in this first row here, and so I want to think about it for a bit. Big P, big T, little t. If I go and I do this, this is the same as this one here with the capital P and the lowercase t. I'm going to basically get the same deal that I got above. If you look right here for this particular box right here, and this one here, what's in there is exactly the same. Okay, so long story short, if I go and I fill in everything and I try to figure out the ratios, I really didn't need a Punnett square this big because I'm going to end up getting ratios that are reducible. And so what I'm going to do already is just go ahead and get rid of this particular row because I already have that kind of gamete here. Column. And I'm going to do the same for this one because I already have this column here where I'm going to get a repetition. Okay. And then what I'm going to do is, because I have the same here and right here, I'm going to get rid of this one. Okay. And I'm going to get rid of one of these because I have I'll get exactly the same results. Right. So what we have left here is basically 4 boxes to tell us what our ratios are going to be. So let's go ahead and complete the remaining squares that are there, which is only again, since we could remove these, it's only these two boxes left. So that's big P, little p, and big T, little t. And then we have big P, and then little t, right? So these are our possible genotypes. And so we see that over here in the first box, we would have gotten 1 out of the 4. That's going to be purple and looks like tall. Are there any other purple and talls? That looks like, so this is purple and tall. We can circle that one. This one is purple and short, so we can't circle that one. And this one is purple and tall, so that looks like 2 out of 4, which is really 1/4, right? So that reduces to 1/4. And the other one looks like it's purple but short. And the other one is also, get rid of that one, the other one is purple and also short. So that again is 2 out of 4 as far as phenotypic ratio goes. So what is our final answer here? Basically we got 2 out of 4 reduces to 1/2. I think I might have said 1/4 a while ago. It's reduced to 1/2. So half, 2 out of 4 is half. So we expect half to be purple and tall and the other half to be purple and short. So the ratio is really 1 to 1. Or we can say 1/2 purple tall and half or 50% purple short in this 2-trait cross. So what are some things to remember from this? What I would remember is if we went back to our monohybrid crosses, which we did not cover here, if you did a monohybrid cross, that would be something like big A, little a, and big A, little a. That would be a monohybrid cross. Your expected ratio would be 3 out of 4 of those boxes would show dominant phenotype, and 1 would be recessive. So you could remember you would get a 3 to 1 ratio there. And when we do an actual dihybrid cross, which would be big A, big B, little b, crossed with big A, big B, little b for your dihybrid, it would be very helpful to remember 9 to 3 to 3 to 1, or 9/16, 3/16, 3/16, and 1. Remember that the 9 would be the dominant phenotype for both. The last one would be the recessive for both, and the middle 2 would be the recombinants, which would show some a variation, dominant recessive, and the other one would be recessive dominant form. So those would be handy to remember for certain types of test questions. So I hope you found this tutorial helpful on dihybrid crosses,